डॉक्टर दिनेश कुमार गौतम है। ये असिस्टेंट प्रोफेसर हैं जूलॉजी के हंसराज कॉलेज दिल्ली यूनिवर्सिटी में। आज ये फूड आइटम्स पे बताएंगे कि क्लासरूम कंडीशंस में बच्चे कैसे कार्बोहाइड्रेट्स को, किस फूड आइटम में कार्बोहाइड्रेट कैसा है, कैसे उसको डिटरमाइन कर सकते हैं, उसके बारे में ये एक छोटे एक्सपेरिमेंट्स दिखाएंगे ताकि कोई भी फूड आइटम हो तो बच्चों को पता चले कि किस फूड आइटम में कितना कार्बोहाइड्रेट है या नहीं है। तो बिना विलंब किए मैं डॉक्टर गौतम से रिक्वेस्ट करूंगा कि अपनी क्लास शुरू करें प्लीज। डॉक्टर गौतम प्लीज।
Okay, thank you, sir. So I am sharing my screen, and please tell me that it is visible to all of you. Sir, is it visible?
हाँ, विजिबल हो गई है। Okay. और आप लैंग्वेज हिंदी अंग्रेजी जैसे भी आपको सुविधा हो आप यूज कर सकते हैं।
जी सर। तो जैसे सर ने बताया था मुझे कि आप जो पार्टिसिपेंट्स हैं दे आर फ्रॉम द schools and both from the government and the private schools, and they are from the 5th to 8th standard. So I have taken a very simple experiment to discuss with you, which we can do in regular classes. I have seen the syllabus also, and the students have to do a simple activities with the topics that are present in their theory classes. Like if they are they are reading about the food or the food components or the digestion of the food that I have seen in the syllabus. So how we can see like what component is present in our food sample and how to test them. So I just start. So as we all know that in food that we eat for energy and to do many different functions, we have carbohydrates, proteins, fats, vitamins, and minerals. And these components have different functions. Like the carbohydrates are used for energy production, like in the form of the glucose, which is used for ATP production, and energy, which is used for doing different works. The proteins, which are present in different parts, or they have many different functions, like they are enzymes to catalyze the reactions, they are antibodies, and many other functions. They transport molecules from one place to other place, like hemoglobin for transport of oxygen in the blood. Similarly, fats we eat, ghee और तेल जो हम खाने में use करते हैं. Vitamins are very essential for our health, and in deficiency vitamin we get different type of diseases. Minerals are also essential, like calcium, iron present in the hemoglobin, or copper or magnesium. We all know the functions of these different food items that we eat. So there are some qualitative tests to detect the presence of sugar or protein in our food samples. There are many different methods are there, and we can test also the lipids or the many other minerals by simple test. But in this today's program, we will do the experiment in which we will see what type of food, what type of sugars or the carbohydrates are present in our various food samples that we eat. So when we are teaching the students in the class about the food or the components of food, and there comes the carbohydrate, and it provides energy. So we can say students that because this food we are eating, like wheat or bread or milk we are drinking, or the juices, fruit juices or fruits we eat, or different other items, or potato, like which particular carbohydrate molecule is present in this food sample, so that we are aware what we are eating and how we are getting the energy from this particular food sample. So these are the requirements. These are very simple, like these tests are very simple, and you can use just test tube and test tube holders or beakers. So not many or very big instrument is required for doing these practicals, and the chemicals are also very easily available in the market. Fehling solution B, Benedict or Seliwanoff, these are available. And in the test sample, we can take various food samples which contain sugars like bread. So you can tell the students to bring some food items from their home. So they can collect 4 or 5 samples each, like bread, potato, milk, chocolate, biscuit, fruit juice, or any other food that they eat. And then they can do the test which sugar is present in which sample. So before that, I just brief about the carbohydrates, that they are aldehydes or ketones of polyhydric alcohols, and also include those biopolymers which yield these compounds or hydrolysis. So we know that we eat the carbohydrates, and they are polysaccharides generally, and then they break down into small molecules like disaccharide or monosaccharides. So we will see these carbohydrates in structure and how they differ from each other. So food samples the students will bring from their home. So the carbohydrates, as I said earlier, that they have the aldehyde or the ketone group. So the carbohydrate जिनमें aldehyde group होता है, they are called aldose sugars. Aldehyde group is CHO, and other sugars which have the ketone group, that is C double bond O. So they are keto sugars. The example of aldose is glucose, and keto sugar is fructose. These carbohydrates are classified into 3 different classes. One are the monosaccharides, which are very simple sugars, and like example glucose and fructose, which are useful to us. And then we have oligosaccharides, in which they have more than 1 monomer unit, like 2 or 10. So depending on the number of monomer units, they are given like disaccharides or trisaccharides or tetrasaccharides. But important one for us are lactose, sucrose, maltose. These are disaccharides. So we generally take these in our food samples, lactose, sucrose, maltose. Now, if the monomer units are more than 10, then it becomes the polysaccharides. And the examples are starch and glycogen and cellulose. So see just the structures of these, how they differ. So the monosaccharides, which are the simplest, and they start with the carbon number 3. They have 3 carbons. So if they have 3 carbon and has the aldose group, so the example is glyceraldehyde. It is an aldose sugar having the aldehyde group and has 3 carbons, so aldotriose. And the other one is the dihydroxyacetone. It is a ketose triose. It has the ketone group C double bond O at carbon number 2 in the picture A. And it is having 3 carbons, so ketotriose. Then other important sugar that we have in our system is 5 carbon, which are the ribose and deoxyribose. As we know, they are present in our RNA and DNA. Ribose in RNA and deoxyribose contains the DNA in the DNA. And we have the ribulose, 5 carbon sugars, which have ketone group. The important ones are the 6 carbon sugars, which have the aldehyde group glucose and the ketone group as fructose. So see the structure of these in B part. The aldose on the left hand side, glucose having 6 carbon, and the aldehyde group is present at carbon number 1, CHO. And in fructose, the ketone group is present at carbon number 2, C double bond O. The rest they are same. So because the glucose has more than 1 number of the hydroxyl groups, so these hydroxyl group can organize or arrange themselves in different orientations, and they form the epimers. Like in the center, it is shown the glucose, and see the position of hydroxyl group at carbon number 2. And when it changes from the left to right side, from the right to left side, it becomes mannose. Carbon number 2, only single change in the hydroxyl group position. If the change occurs at position number 4, then it becomes the galactose. So galactose is also important because it is present in lactose, which is a disaccharide and present in milk. So these are the epimers of glucose. Now this glucose can be in a open chain structure, or it can form a ring form structures. In the first, it is the open chain structure. Now the aldehyde group at carbon number 1 forms a bond with the hydroxyl group at carbon number 5 and forms a ring-like structures. And because of this, we have two different forms of the glucose. One is the alpha form and other is the beta forms. So in alpha form, the hydroxyl group is present below the perpendicular axis, and in beta form, it is above the axis. So this is how they differ from each other. This is the structure of the glucose and fructose in the ring form. So at the top, it is the alpha form having the hydroxyl group at carbon number 1 below in both the sugar, and in the next one is the beta form where the hydroxyl group is present above. And these are the structures derived from the normal natural molecule like pyrin and furan. So the next one is oligosaccharides, where we have the example disaccharides having 2 monomer units, and the maltose has 2 glucose units, 2 monomer units. Lactose has glucose and galactose, and sucrose has glucose and fructose. See how this maltose is formed. So at the top, we have 2 glucose units which are separate. So when they come close, they form the linkase, glycosidic linkase between the carbon number 1 hydroxyl group and carbon number 4 hydroxyl group. So these two combine and release 1 molecule of water. So hydrolysis, the condensation occurs, H2O is released, and these 2 sugar units, molecule, the glucose, are joined with a glycosidic linkase. So if 2 glucose molecules are present, then it is a maltose disaccharide. See in this picture, we have shown the lactose at the top. In lactose, we have the glucose and the galactose. See on the right hand side, it is the glucose in beta configuration. Hydroxyl group is above, and on the left hand side, it is the galactose. See the position of carbon, the hydroxyl group at carbon number 4, it is above. So it is the galactose, and they join with the hydroxyl group at carbon number 1 of galactose and hydroxyl group of carbon number 4 in glucose. So this is alpha 1,4 linkase between the glucose and galactose. The disaccharide becomes the lactose. What happens in case of the sucrose? It is disaccharide made up of glucose and fructose. See on the left hand side, it is glucose. So, and on the right hand side is galactose, fructose. And they see the bond between the glucose and fructose. It is between carbon number 1 hydroxyl group and the hydroxyl group at carbon number 2 in fructose. So the carbon number 1 in the glucose is the aldehyde group, and carbon number 2 in fructose is ketone group. So both these aldehyde and ketone group are involved in bonding. So in sucrose, there is no free aldehyde or ketone group. And later we will see that because of this, we have a sucrose, which is a non-reducing sugar. So these are the in these structures, which are important thing that you have to tell about this, tell to the students that how these different sugars differ. And on the basis of these, they will perform different tests. Now we have the polysaccharides, which have more than 10 monomer units. Examples are starch and glycogen. The starch is present in plants. It is formed by photosynthesis. Glucose is formed. Then these glucose units combine together and form a large the polysaccharide, which is called starch. And in animals, it is the glycogen, which is stored in different cells, especially the hepatocytes or muscles. So these are polysaccharides. Now the structure of the sucrose, the starch. It has two components. One is the amylose and other is amylopectin. And in this, the glucose molecules are joined together with alpha-1,4 glycosidic linkase. See the one end at the top. It is on the left hand side. It is non-reducing end because there is no aldehyde group present. At this place, it has the HOH, hydroxyl group at carbon number 4. And on the right hand side, see, if no sugar is attached and it is the free carbon, carbon number 1, it will have the aldehyde group of the sugar, glucose, free. So it is the reducing end, and on the left hand side, it is the non-reducing end. And these sugars are joined with the alpha-1,4 glycosidic linkase. This is a linear structure, but this starch forms a branch chain also. And branching occurs by forming a glycosidic linkage between the alpha-1 hydroxyl group and the hydroxyl group at carbon number 6 in the glucose molecule. So on the left part, the B diagram shows this linkase, alpha-1,6 glycosidic linkase. And these two components, amylose and amylopectin, are arranged to form this
आपकी वॉइस नहीं आ रही है गौतम जी। आप अपने आप को अनम्यूट कीजिए। अपने आप को अनम्यूट कीजिए।
ओके सर नेटवर्क एरर था तो दोबारा ज्वाइन किया इसलिए।
नहीं कोई बात।
ओके सर। So the carbohydrates are of two types. One are reducing and other are non-reducing sugars. The reducing sugars are those which have free aldehyde or ketone group, and they can reduce, convert Cu2 plus to Cu plus. For example, in monosaccharides, we have glucose and fructose. You have seen the structure of glucose and fructose, and if they are single molecule in monosaccharide form, they will have a free, the aldehyde and ketone group. Now come to the disaccharide in maltose and lactose. We have seen the bond that is formed between the two monomer units is 1,4 linkase, alpha 1,4 glycosidic linkase. One molecule is one aldehyde group is involved in the linkase, but the other aldehyde group on the right hand side is free in both maltose and lactose. So they can also convert the Cu2 plus to Cu plus. So they are also the reducing sugars. Now the non-reducing sugars, which do not have the free aldehyde or ketone group and which cannot convert Cu2 plus to Cu plus. The example is disaccharide, as we saw in the diagram or the picture, the structure of the sucrose, where both the aldehyde of glucose molecule and the ketone group of fructose are bound together, are involved in glycosidic linkase formation, and they have no free aldehyde or ketone group. So they are the sucrose is a non-reducing sugar. So depending on the free aldehyde or ketone group, they will be performing these tests. And this is how we can detect whether a sugar is reducing. Reducing means it is either the glucose, fructose, or maltose, lactose, or if it is giving no color, then it is a sucrose. So now coming to the tests, there are different tests which can be performed, like a Molisch test. It is a general test for carbohydrate, and this is to be done to check whether the sample that we have is a carbohydrate or not. It can be plain water, or any protein solution can be there. So it is a first test to confirm that the sample is a carbohydrate. Then we can perform the test of fat.
आपकी आवाज नहीं आ रही अभी। दिनेश जी, आपकी आवाज चली गई। अपने आप को अनम्यूट कीजिए।
And then we can in the last do the iodine test, which is for starch, a polysaccharide. So the Molisch test, as I said, that it is a general test for all carbohydrates. All carbohydrate will give, and it is to check whether the sample is a carbohydrate or not. And the principle is that the concentrated H2SO4 hydrolysis glycosidic bond to yield monosaccharides, which in the presence of an acid gets dehydrated to form furfural and its derivatives. And then these products react with the sulfonated alpha naphthol to give purple or violet complex. This is a very simple test and takes just few minutes to perform. So can be done anytime in the lab. This is the reaction that the glucose, if it is a carbohydrate like glucose, so H2SO4 will do hydrolysis. So the water will be removed, dehydration will be there, and forms the hydroxymethyl furfural. And in presence of the alpha naphthol, they form a condensation product which has a purple or violet color ring. How to do this test? The procedure is take 2 ml of test sample in a test tube, add 2 to 3 drops of alpha naphthol solution to it and mix. Then very gently add concentrated H2SO4 along the side of the test tube so that two distinct layers are formed. So when you are adding H2SO4, tilt the test tube at a angle like 45 or 60 degree angle, and add the H2SO4 drop by drop so that it goes slowly into the tube, and then it forms two distinct layers. Then the reaction will be there, and then observe the color at the junction of the two solutions. So appearance of a purple or violet color ring indicates the presence of carbohydrate in the test sample. So see, the ring is formed at the junction of two different solutions. So this indicates a positive test. If the sample is giving a positive test, then we can perform other test, that is whether the sugar is a reducing sugar or non-reducing sugar. And for this, the test is Fehling test. And in this test, copper sulfate present in Fehling solution or reagent forms cupric hydroxide, which on heating forms cupric oxide. The reducing sugar reduces the cupric oxide, that is Cu plus, and forms cuprous oxide having red PPT. So we have to observe the formation of this red PPT in the test. This is the chemical reaction. Copper sulfate plus potassium hydroxide, they form cupric hydroxide. When it is heated, it forms cupric oxide, CuO. This CuO will react with the glucose or other sugar molecules and forms cuprous oxide, which is red in color. So by observing the red color in the precipitate, we can say that the test is positive and the sugar is a reducing sugar. So take 1 ml of the test solution in a test tube. Take 0.5 ml of Fehling solution A and 0.5 ml of Fehling solution B. Mix them well. Hold the test tube firmly in a test tube holder and keep the test tube in a vigorously boiling water bath for 2 minutes. So within 2 minutes, you will observe the color change. So the test is very simple. You require just 1 ml of the reagent and 1 ml of the test sample, and the result you will get in 2 minutes. See, this type of red color will be shown by the glucose and fructose, which are reducing sugars, and the sucrose, which is a disaccharide, is not showing any color, showing the same, the blue color of the Fehling test, so reagent. The other test can be done for reducing sugar is the Benedict test. It is more convenient than Fehling test, and the reagent is more stable. And in the test also, the presence of reducing sugar results in the formation of red PPT of cuprous oxide. The reaction is almost same, just the chemicals are a little bit different. So the reaction is that Na2CO3 forms NaOH. This NaOH reacts with copper sulfate and forms the cupric hydroxide. On heating, this forms cupric oxide. And this cupric oxide reacts with the glucose and forms the cuprous oxide. The glucose is taken as an example. The other sugar can also be there, like fructose or any disaccharide. The red color will be there in the last, after 2 minutes. So add 0.15 or 1 ml of test solution to 2 ml of Benedict reagent, and then mix them and keep it in boiling water bath for 2 minutes. And observe the formation of red PPT. So in the last, you will get this red type of precipitate, which indicates that the test is positive and the sugar is a reducing sugar. Now, you have done the test for reducing or non-reducing. Now the sugar can be a disaccharide or monosaccharide. Both these give the positive test for reducing sugar, Fehling or Benedict. And you have to differentiate whether the sugar is a monosaccharide or disaccharide. So we can do this Barfoed test. The test will be positive for the monosaccharides. So monosaccharides will react in just 1 or 2 minutes and give the color. Whereas if disaccharide is there, so when you are keeping the test tube in a boiling water bath, so it will hydrolyze and form the monosaccharides. Like the lactose will break down into glucose and galactose, and then they will form the color reaction product. So depending on the time, whether the color comes within 1 or 2 minutes, it is a monosaccharide, and if it takes more time, like 7 or 8 to 10 minutes, then it is a disaccharide. So you can distinguish between the monosaccharide and disaccharide by doing this test. This occurs in a acidic medium like the cupric acetate, and form cupric hydroxide, which on heating forms the cupric oxide, and it reacts with the sugar molecule and form the cuprous oxide with a red PPT. They take 2 ml of Barfoed's reagent in a test tube and add 1 ml of test solution and mix it well and keep in a boiling water bath. So if it is in 1 or 2 minutes, it is a monosaccharide. If it takes longer, then it is a disaccharide. So this type of red PPT will form at the bottom of the tube. Now, till now we have done that the sugar is reducing, and suppose it is a monosaccharide. So the monosaccharide can be again the fructose or the glucose. So this test is done, Seliwanoff test is done to find that the sugar is a aldose or the ketose. So this test is positive for ketose sugars. So if test is positive, that means the fructose is present in the sample, or if the test is negative, then it shows that the sugar is a aldose sugar and it is naturally glucose. So ketoses undergo dehydration to give furfural derivatives, which then condenses with resorcinol and form a red complex. Now, in this case, if we are using the disaccharide like sucrose, so and keep the solution for long time, like 10 or 15 minutes. So naturally the sucrose will hydrolyze into glucose and fructose, and then it will also give the positive test. So again, the time is more important to note down in this test, that immediately it will give the color, then it is a ketose or the fructose. So because this test is positive for ketose sugar, so we have taken the fructose as a sample. So the reaction is between fructose, and we are using concentrated HCl. So dehydration will occur, water molecule will be removed. It forms the hydroxymethyl furfural. In presence of resorcinol, they form a condensation product, which is having red color. So add 1 ml of test solution to 2 ml of Seliwanoff's reagent and warm in a boiling water bath for 1 minute. And note the appearance of a deep red color. So these sugars like fructose will give this type of color. And if it is not, then it will not show this red color, the glucose or the aldose sugars. Now for to confirm that the sample we have got is a sucrose, which is a disaccharide, and it is showing a negative Fehling or the Benedict test, we can do this test, hydrolysis test for non-reducing disaccharide. That is, we will keep the sucrose in a acidic medium so that it is hydrolyzed and forms the glucose and fructose monomers. And then later we can do the Benedict test or Fehling test, which will be positive in the solution. So this is the chemical reaction that sucrose molecule. See in this the aldehyde group at carbon number 1 in glucose and the ketone group in the fructose are bound together, and there is no free aldehyde or ketone group, so the sugar is a non-reducing sugar. But when we do the hydrolysis in acidic condition, it breaks sucrose into glucose and fructose monomers. And if we perform the Benedict or Fehling test, it will be positive, that the reducing sugars are present in the hydrolyzed sample. So take 2 ml of test solution in a test tube. Okay, so take 2 ml of test solution in a test tube and add 2 to 3 drops of normal hydrochloric chloric acid, HCl. Boil for 5 minutes and cool. In these 5 minutes, it will be hydrolyzed and form the glucose and fructose. Then to neutralize the acidity HCl, we add the NaOH. Then we perform the Benedict or Fehling test and observe the formation of red PPT. If it is hydrolyzed, then it will give the same Benedict colors. The red PPT will be there after the test. For polysaccharides, we can do the iodine test, which is positive for starch. Generally, we take the starch from the plants, and the iodine test is a very simple test. You have seen the structure of the iodine, the starch, that it is made up of the amylose and amylopectin. So what happens? This iodine molecule get adsorbed within the branching of the amylose and amylopectin, and because of this, it gives a blue color. There is no chemical reaction as such, but this color is developed because of adsorption of this, or the adsorption of this iodine within the branches of the iodine, the starch, amylose and amylopectin. So chemical reaction is starch plus iodine will give blue-black color complex. The test is very simple, that take a 1 ml of test sample in a test tube and add 2 to 4 drops of iodine solution, and then mix well and note the change in color. So the positive test will be showing a blue color, dark blue color, and the negative test shows the same yellow color of the iodine. So we can see that the sample is a polysaccharide or starch. So when the students perform all these tests from their food samples, so you can make a table like this, observation table, where you have the test name and the sample A, B, C, D, E are these the food samples, like the wheat or the milk, or your potato or the fruit, biscuits or chocolates, or whatever sample they have got from the home. And then they perform the test and indicate in the table that it is positive plus sign. And if it is negative, then it is a negative sign. So the example is shown in the A part, that it is positive for mollusk, means it is a carbohydrate. Then it is showing positive for Fehling test, that means it is a reducing sugar. Then it is performing positive Barfoed test, and then it means a monosaccharide is there. And it is negative for the Seliwanoff test. That means it has an aldehyde group, because if ketone group hota to ye positive hota, but it is negative, so it is a aldose sugar. Because it is a monosaccharide, so there is no need for the hydrolysis test. And similarly, it is a monosaccharide, so no need to do the iodine test. And in the last, we can confirm that because of these tests or the groups present in the sugar molecule, it is a glucose. And similarly, you can go for other samples B, C, D, and E. So we can find that this particular food sample contains this particular sugar. So students will be excited to see that what they are eating when they eat mango. So which sugar are they taking in the mango? Or if they are eating the apple, what sugar is present in apple? Or if they are drinking the milk, what sugar is present in milk? So these are the simple experiments which can be done in the classes like 5th to 8th, and these are very simple tests, easily done. So in the result, you can write that the given food sample shows positive or negative depending on what test it has been done. The following tests, the name of the test, whether it is positive or negative, you can write there, and then you can confirm that the given food sample contains or not contains the particular sugar, like it contains glucose, or it contains fructose, or it contains lactose, or maltose, or sucrose. So these are the observations and the results which can be shown to the students, or they will, after they will write about the results in the notebook. These are the precautions that can be done, like use clean and dry test tubes for individual test, and use separate pipettes or droppers to add reagents. Otherwise, if you are taking the same dropper to take different samples, then they will be mixed, and the reaction can, or the chemical the response or the colors may be different, and then the positive test, it has to be. Use acids carefully during test so that it is not spilled on someone else, or use test tube holders to boil the solutions in the tube. Do not keep the test tube in hand while boiling or keeping in the boiling water bath, and keep adequate distance from burner while boiling the reagents, so that when boiling occurs, and if you are keeping the test tube for long period of time, that then the drops can come out, and it may have the effect on the skin. These are some precautions. These are the references from which this has been taken. You can see some YouTube videos also, and get how these tests are actually done. So this is all about from my side. So thank you for listening, and now you can ask some questions if you have.
थैंक यू डॉक्टर दिनेश। सबसे पहले मेरा क्वेश्चन टीचर्स के लिए है कि ये जो अभी डॉक्टर साहब ने दिखा दिया, बायोकेमिस्ट्री बता दी, जो भी शुगर्स है, कॉम्प्लेक्स शुगर्स है या सिंपल शुगर्स है, बट द बेसिक पॉइंट इज कि एक्सपेरिमेंट्स में आपके स्कूल्स में मतलब आप लोग के फैसिलिटीज हैं, या कैन यू जनरेट द फैसिलिटीज जो 7th या 8th के स्टूडेंट्स को आप लोग ये सिंपल एक्सपेरिमेंट्स दिखा सकते हो? क्योंकि जब तक बच्चे को दिखाएंगे तो सीइंग इज बिलीविंग, तो उसको कैसे समझ आ जाएगी? तो ये मैं चाहूंगा कि टीचर्स जितने भी हमारे साथ है, कोई हमें बता दे कि इसमें से कौन प्रैक्टिकली आप लोग क्लासरूम में दिखा सकते हैं।
Good morning, sir.
Good morning.
Sir Arjun Chatterjee from Bal Bharati Indraprasth Yojana Vihar.
Yes.
11th and 12th, in their practicals, they have to actually test for sugars using Benedict solution, Fehling solution. So the reagents are already there. What we can do is, while the students are performing those tests, we can just call the 6th and 8th students. They can observe. They might not want to work with boiling substances, but while the older students are doing that, they can observe. So they appreciate the color.
For your observation only. That's not that the student has to perform the test by himself.
So
but
it is
teacher has to show.
Yes, sir, because boiling water and burners, etc., are required. So maybe class 8th, maybe, but class 6th and 7th, they need closer supervision, because I wouldn't want them to handle boiling substances, burners, etc.
डॉक्टर साहब ने, डॉक्टर दिनेश ने सही बताया कि एक टीचर दिखा के करेंगी। ये बोल रहे हैं कि बच्चे को बोला जी अपना फूड आइटम ले आइए, जो भी आप खाते हो, चाहे बिस्किट है चाहे कोई, वो टीचर डिमॉन्स्ट्रेट करेगा कि देखो इसको, ये आप जो खा रहे हो इसमें सिंपल शुगर है या इसमें कॉम्प्लेक्स शुगर है या क्या है। दैट्स व्हाट ही वाज टेलिंग। क्योंकि उससे क्या होता है कि इंटरेस्ट एक बच्चे में भी जनरेट हो जाएगा कि मैं जो बिस्किट खा रहा हूँ कि इसमें जितनी भी शुगर है वो मोनोसैकेराइड्स है, पॉलीसैकेराइड्स है, ओलिगोसैकेराइड्स है, पचेगी नहीं पचेगी, बिकॉज सुक्रोज पचता नहीं है। तो इसी टाइप का बेसिकली इंटरेस्ट, दैट्स व्हाट, उसको पता है कि मैंने एप्पल खाया तो इसमें ये वाला शुगर मेरे को मिला। पर्पस इज दैट कि जो भी टीचर एक्सपेरिमेंट सॉर्ट ऑफ मतलब डिमॉन्स्ट्रेट कर सकता है क्लासरूम में, बच्चे को इन्वॉल्व करके, उसको बच्चे को तो एक्सपेरिमेंट नहीं करना है, लेकिन टीचर क्लासरूम में दिखाएगी या दिखाएगा कि वो देखो आपने आज एप्पल खाया तो एप्पल का सॉल्यूशन बना के इसमें कौन सा शुगर है।
Absolutely, sir.
अरे बच्चे थोड़ा एक्साइटेड फील करते हैं व्हेन यू इन्वॉल्व देम। ये छोटे बच्चों को जैसे बोलो कि घर से आप थोड़ा सा अपना कोई भी जो खाते हैं वो लेकर के आइए, तो बच्चे यदि एक बच्चा कोई दूध ले आता है थोड़ा सा 4-5 ml, या फिर कोई बच्चा अपना कोई फ्रूट ले आता है या कोई और ब्रेड ले आता है, तो दे फील एक्साइटेड कि आज हमें कुछ नया करना है। जब क्लास में नया देखते हैं, घर जाके बताते हैं कि हमने आज ये किया और हमें ये वाली इस सैंपल में ये वाली शुगर है, तो उनको बड़ा अच्छा लगता है अंदर से, और कुछ नया करने का उनका एक अंदर से आता है कि हम ये भी करें या ये सीखें और कुछ इससे डिस्कस करें और आगे इससे। यही
Yes,
है कि
sir.
बच्चे थोड़ा एक्सपोजर हो उनका।
Thank you, sir. Definitely try that. Thank you.
और किसी टीचर का क्वेरी हो तो डॉक्टर साहब जवाब देंगे। सब लोग साइलेंट बैठे हैं, मतलब उसमें मजा नहीं आता।
Good morning, sir.
गुड मॉर्निंग।
Myself Priyanka Kaushik. Basically, most of the government school don't have science lab. We have provided only a science kit. So it is very difficult to show this kind of experiments in the classroom. Because we have only few equipments are there. Only equipments, not the reagents like ये Fehling solution, Benedict solution, Barfoed, ये सब हमारे schools में available ही नहीं है in the most of the schools. So कल जो मैम ने कराया किसी extent तक वो हम लोग फिर भी कर सकते हैं, लेकिन ये तो बहुत ही, अभी तो हमारे schools हैं अभी grow कर रहे हैं infrastructure level पे, so it will take few years or maybe then we can do. Thank you.
आपके साइंस किट में क्या रहता है?
साइंस किट में थोड़े बहुत टेस्ट ट्यूब, माइक्रोस्कोप, स्लाइड्स और ये 1 year कैलिपर्स, समथिंग लाइक ऑल दीस थिंग्स।
तो अभी यहाँ पर एक सर ने बोला कि वो 11th में कराते हैं, तो यदि आपके स्कूल में अवेलेबल है तो आप बच्चों से फूड आइटम्स मंगाइए और उनको वो बच्चे करके दिखा दें जो लैब में 11th और 12th के बच्चे हो, तो डिमॉन्स्ट्रेशन में भी थोड़ा सा फर्क पड़ता है बच्चों को कि हमारे सैंपल में हम जो सैंपल ले गए उसमें से टेस्ट किया आज हमने। तो थोड़ा एक्साइटमेंट फील होता है बच्चों को।
जी, थैंक यू।
और किसी का ऑब्जर्वेशन?
बट सर वो जिन सर ने अभी बताया कि वो 11th क्लास में कराते हैं, हमारे तो स्कूल ही 8th तक के हैं। Neither we have space nor equipments.
नहीं वो ठीक है, that's correct. लेकिन साइंस किट्स वगैरह हैं आपके इधर?
Yes, we are having kits जिससे हम डिस्प्ले कर सकते हैं उन्हें कि ये वर्नियर कैलिपर्स है, माइक्रोस्कोप है जिनसे हम दिखा सकते हैं।
Okay, see what we will do then कि चूंकि किट्स आपके पास ये है तो हम कल के जो लेक्चर है we will orient it in such a way कि जो आपके पास साइंस किट्स पड़े हुए हैं उसको आप कैसे यूटिलाइज कर सकते हैं बच्चों को दिखाने के लिए और उनको समझाने के लिए।
That will be better for us क्योंकि we have a very less facility to demonstrate such experiment. We have nothing.
नहीं, दैट्स यू
Like
आर
these
करेक्ट,
things, है ना?
क्योंकि तभी तो हम आपसे जनरेट करने की कोशिश कर रहे हैं कि
जी।
आपने बताया तो हमें पता चला कि क्या
जी
फैसिलिटी अवेलेबल है,
जी
तो
जी
अकॉर्डिंगली इसको हम डिजाइन कल के लेक्चर्स में इस तरीके से करेंगे कि आपके पास जो फैसिलिटी है, साइंस किट्स है, ये उसको कैसे आप ऑप्टिमली यूटिलाइज कर सकते हो और बच्चे को सिखा सकते हो या समझ दिखा सकते हो कि देखो ये चीज ऐसे देखी जाती है, इसमें ये देखा जाएगा.
that will be more useful for all of us.
Yes, we will be doing that. Rather, एक मैं थोड़ा और रिक्वेस्ट करूंगा आपके जो 5th to 8th में, क्योंकि 90% आप लोग गवर्नमेंट से टीचर्स हो, तो आपका जो सिलेबस है अगर व्हाट्सएप पे डाल दो तो एक थोड़ा सा ओवरव्यू ले लेंगे हम कि उसमें से आपकी जो साइंस किट को कैसे हम यूटिलाइज करें.
Sure, sir. We will do that.
तो वो
Okay.
आप व्हाट्सएप पे डाल देना, तो वो हमें थोड़ा सा हमारे एक्सपर्ट्स के लिए आसानी होगी.
Okay, sir.
सर हम लोग ना
स्पेशली
हम इंडेक्स की पिक लेके डाल देंगे उसमें जो भी
हाँ जी,
chapters
इंडेक्स ही खाली चाहिए. यू डोंट नीड इनसाइड एनीथिंग, इंडेक्स आप डाल दो खाली, तो एक हमारे लिए भी आसानी होगी उसको डिजाइन करने को, ताकि आप जो भी हमारे साथ इतने जुड़े हो एक प्रोडक्टिव सेशन हो जाए.
जी सर.
और किसी
सर
का
मुझे
कोई, हाँ जी
सर मुझे एक चीज पूछना था ना कि ये जो साइंस किट बता रहे हैं इसमें क्या क्या मटेरियल होता है वो भी पता चल जाए ना, जैसे कौन कौन सी स्लाइड्स हैं जो ये इजीली दिखाते हैं अपने स्कूल में बच्चों को, ताकि उसके बारे में थोड़ा सा हम ऐड कर पाए साइंस किट में जो चीजें हैं.
देयर सर बिल्कुल इंफॉर्मेटिव लेवल का मटेरियल होता है हमारे पास कि हम बच्चों को शो कर पाए कि ये स्लाइड है, ये माइक्रोस्कोप है, इसको हम ऐसे यूज कर सकते हैं, ये लेंसेज है, ये कॉन्वेक्स है, मतलब जस्ट बिल्कुल सिंपल बेसिक थिंग कि बच्चों का एक्सपोजर हो जाए. बस सर इतना ही है हम लोगों के स्कूल में.
ओके, आप जैसे अमीबा की स्लाइड्स वगैरह दिखाते हैं, अमीबा या और कोई सेल टाइप?
सर पॉसिबिलिटी ही नहीं है.
माइक्रोस्कोप तो आप दिखाते हैं ना बच्चों को कि माइक्रोस्कोप है कैसे यूज करते हैं.
So you can take that microscope as a dummy one. Most of them is not working.
ओके. ठीक है, आप शेयर कीजिए फिर देखते हैं.
10x ये तो लेंस तो रहता ही होगा ना.
यस सर, यस सर.
तो अगर नीचे लीफ रखे तो वो थोड़ा सा उसके वो आदमी देख ही सकता है ना थोड़ा बहुत.
जी
या
जी
कोई मतलब ये कोई भी मतलब छोटा पीस हो जो उसको थोड़ा मैग्नीफाइड दिखेगा उसको.
सर प्याज की झिल्ली भी दिखा सकते हैं.
Exactly, sir. We can do that.
ओके, तो ठीक है, वी विल इसको हम, आप हमें सिलेबस भेजिए, हम इसको रिडिजाइन करेंगे कल के लिए. अभी
ओके सर.
ठीक है ना.
जी
तो अभी और किसी का कोई ऑब्जर्वेशन है, कुछ ऐड करना है तो काइंडली बताइए, बिकॉज़ देन वी विल हैव अ ब्रेक ऑफ 10 मिनट्स. तो तब तक हमारे डॉक्टर साहब आ जाएंगे, तो वी विल स्टार्ट एट 11:10.
Am I audible? Good morning,
Yes.
sir.
Yes, yes, ma'am.
Shobna Sharma. I just want to ask, is there any WhatsApp group where the teachers are connected?
Yes, there is a WhatsApp group which is already connected.
I am not connected in that group. And I have one suggestion. If what is going to happen in tomorrow's session, if we know that what is the topic, so it will more helpful for the
Yes.
professors who are taking. Because I am seeing that we have a very learned professor with us. They are from the Hansraj College, so it is our pleasure to be with them. But the thing is, there is a missing connect between, because grade 5 to 8 is very low level for them. So the same problem which teachers are facing.